Hello everyone. My name is Dr. Vinod Shinde, Assistant Professor, Department of Microbiology, Shankarrao Chavan Mahavidyalay, Ardhapur, District, Nanded. Today we are going to discuss about the Unit 1, Air Microbiology, which is in the syllabus of BSc second year paper Applied Microbiology. In this chapter or unit, we'll discuss about the definition of air microbiology, the composition of air, number and kinds of microorganisms in air, significance of microorganisms in air, then enumeration of microorganisms in air and how to control the microorganisms in air. Before that, we'll discuss about the summary of this topic that contains what is air or what is the role of air in the day-to-day life. Air is ubiquitous in nature. It occupies the atmosphere, interparticle space in soil and everywhere. But however, it does not provide necessary nutrients, proper physiological conditions, as well as physical support to the microorganism. Hence, it is not the natural habitat for the growth and reproduction of microorganisms or any other living being. The gaseous content of air are quite static, and any changes in it may cause harmful effect on living world. Air at a particular site contains variety of microorganisms including bacteria, yeast, molds, viruses, etc., which are carried from different sources. It may contain pathogens for plant, human beings, and other animals, which causes different diseases. Infection of crop on field by airborne pathogen results to a great economic loss to the farmers and ultimately to the nation. Hence, pure air free from any extraneous physical, chemical, or biological agent is very essential for healthy life of plant and animals. The microorganisms in air also causes contamination of pure cultures in laboratories and industries, which create harmful effects and endospores of bacillus vegetative cells and other bacteria, cells of other bacteria, and asexual spores of molds are the common airborne contaminants. Microbiological analysis of air can be performed by blowing a known volume of air onto solid or liquid growth medium, which we will discuss in the enumeration of microorganisms in air. And a removal of microorganisms or killing of microorganisms in the air is called air sanitation. Air sanitation, which we will discuss in the control of microorganisms. Various physical and chemical means are used for the air sanitation purpose. The use of UV radiation, ultrafiltration, gaseous sterilizing agents, and some commonly used methods for air sanitation. The gaseous agents are particularly used to sterilize enclosed rooms and machineries in the dairy, pharmaceutical industries, pathogenic laboratories, hospital wards, and operation theaters. Each of these means have particular mode of action by which it kills microorganisms and a spectrum of antimicrobial activity. Each of these has some significant property as well as drawbacks on, and no one is ideal for air sanitation. Air is free, air free of dust, microorganisms, chemical pollutants, and with normal composition of gases is very essential for health of human beings, animals, and plants. Addition of any physical, chemical, or biological agent in air or changes in the concentration of normal constituents harmful to the health of plant, animals, and other living worlds is called air pollution. that that is threatening problem in the world. Now we'll discuss about the definition of air microbiology. Air microbiology deals with the study of microorganisms present in the air that have harmful as well as beneficial significance with regards to human. As we have discussed, air is not a natural environment for growth and reproduction of microorganisms. It does not contain necessary amount of moisture and also kinds of nutrient in the form utilizable by bacteria and other microscopic organisms. Therefore, air is of considerable importance economically and in the public health. The atmosphere consists of mixture of gases. The composition of air is very much important in this regard. Various layers are recognized in the atmosphere up to the height of 100 kilometers. The layer nearest to the Earth is called troposphere. In temperate region, the troposphere extends up to 11 kilometers, whereas in the tropic up to 16 kilometers. This troposphere is categorized by load of microorganisms. The atmosphere consists of various mixture of gases, moisture, and solid particles. According to Landsberg in 1951, the composition of air is as shown, that is, nitrogen is 78.03, oxygen is 20.99, argon is 0.94, carbon dioxide is 0.03, hydrogen is 0.01%, neon 0.0012%, krypton 0.0010%, helium 0.0004%, xenon 0.0001%, and ozone, water vapors, and dust particles are in the variable amount in the atmosphere or in the air. The amount of water vapors, dust particles varies greatly with different factors such as time of day, season of year, wind current, and height of the atmosphere. The concentration of gases in the air is maintained constant by metabolic activities of plant, animals, microorganisms, and all other living beings. Addition to any physical, chemical, and biological agent causes air pollution. Any changes in the normal concentration of gases in the air is also harmful to the living world. Air is the basic need of life. Gases in air plays a significant role in growth of living organisms. Among the different gases, the most important is oxygen, which is well known as pranavayu or the gas for life or oxygen. Respiration is one of the most widely used mechanism of ATP generation in living organisms. Oxygen acts as ultimate electron acceptor in aerobic respiration. Carbon is the most abundant element in cellular biomolecules, contributing about 50% of cell's dry weight. The primary producer, that is plant, algae, and photosynthetic bacteria, uses carbon dioxide as the sole source of carbon for biosynthesis of cellular organic compounds. The heterotrophic microorganisms also utilizes carbon dioxide in small amounts for biosynthesis of organic compounds, which is mainly provided by catabolism of organic compounds. The atmospheric nitrogen acts as a source of nitrogen elements for nitrogen fixing microorganisms and their host plants. Proteins, nucleic acids, and other nitrogenous biomolecules of the cell are synthesized using this source of nitrogen. Hydrogen acts as a source of electron for hydrogen oxidizing bacteria. Ozone plays important role in filtration of harmful UV radiations of sunlight. Dust particles act as physical support to the microorganisms, and humidity of air affects the existence of microorganisms. Microorganisms play important role in utilization and synthesis of gases and to maintain their concentration. constant in the air. The microorganisms vary in the number and kinds in the air. The number of microorganisms in the air is dependent upon the activity in the environment and upon the amount of dust stirred up. An active environment shows a higher bacterial count than a less active one. The number in dirty, untidy rooms is greater than in the clean rooms. Marine air also contains microorganisms, but although it is to the lesser extent than in the terrestrial air. Kelly and Lane in 1957 collected air samples over the Atlantic Ocean at altitude of 8,000 to 9,000 feet. Species of following genera were isolated by them, that is Achromobacter, Bacillus, Corynebacterium, Flavobacterium, Micrococcus, Sarcina, and Staphylococcus. The investigators concluded that the organisms from marine air were similar in kind to those found over the land. As also discussed, air is not suitable environment for the growth and reproduction of microorganisms. It does not contain necessary nutrient and proper physiological conditions. Air is a transient place for the occurrence of microorganisms. That is, they stay in air territory. The microorganism in air may travel in the horizontal direction up to the height of 20,000 feet or more with the air currents. Pollen grains and dust have been recovered at the high altitudes and indicates the importance of air as a vehicle for the transmission over a wide area. The kind and number of microorganisms in air vary depending upon the source from which they are carried. The air above a bare surface contains more organisms than the air above the land covered with vegetation. Air over marine environment also contains microorganisms, as discussed, although to a lesser extent from the terrestrial air. Organisms attached to air dust particles or in the droplet of moisture settle down at a much faster rate than the free vegetative cells or spores. A damp or humid atmosphere contains fewer organisms than the dry atmosphere. Therefore, air during summer seasons contains many more organisms than during the winter or rainy seasons. In general, aerosols of microorganisms lose viability very rapidly. Asexual spores of fungi, which commonly occur in air, are the sporangiophores related from the sac-like structure called sporangium. Conidiospores, which arise in chain at the tip of hyphae. Chlamydospores, thick-walled single-celled spores, which are highly resistant to adverse conditions, and the blastospores formed by budding of mycelium. The number of microorganisms in the air depend on the activity of the environment and the amount of dust stirred up. Microorganisms stay in the air for varying period of time depending on the speed of air current, humidity, sunlight, temperature, size of particle on which they are attached, and the resistance to organism to temperature, resistance of organism to temperature, radiation, and other factors of environment. All living microorganisms, including bacteria, molds, yeast, viruses, etc., are sensitive to UV radiations of sunlight. Spores are more resistant than vegetative cells. Certain molds and yeast are common in air, and in some localities they may dominate the bacteria. Spores of molds and yeast are quite resistant to light, which is rather lethal to bacterial cell as well as spores. Another reason for the greater prevalence of fungal spores than bacteria is that the reproductive process of fungi releases them in dry state into the air with tremendous number, while bacteria multiply in moist places from which they do not readily escape. In bacteria, sporulation is the mechanism of survival but not the reproduction. Spore of phytopathogenic fungi are released in large number from the fields of infected plants. The microflora of air consists chiefly of spores of fungi, especially Alternaria, Aspergillus, Cladosporium, Fusarium, Geotrichum, Homodendricum, Dendrum, and Penicillium. Ascospores of yeast, Conidia of Streptomyces, and endospores of Bacillus and Clostridium. The species of Bacillus are relatively abundant and usually act as airborne contaminants of pure culture in microbiology laboratories, other research laboratories, hospitals, and in the industries as well. The best known member of this group is Bacillus subtilis. It is known as hay bacillus and is one of the most common bacterium found in nature. It is natural. Its natural habitat is the soil and vegetation. Since it is a spore-forming organism, it is very resistant to drying and other unfavorable environmental conditions. The other species of Bacillus include Bacillus cereus, Bacillus thuringiensis, Bacillus anthracis, Bacillus megaterium, Bacillus licheniformis, Bacillus polymixa, etc. They are gram-positive, spore-forming motile rods. The other spore-forming bacteria which may found in air are the species of Sporolactobacillus, Desulfatomaculum, and Sporosarcina. The only spore-forming cocci appear in tetrads or packets is Sporosarcina. Among the non-sporulating bacteria, species of Alkaligens, Corynebacterium, Flavibacterium, Micrococcus, Sarcina, and Staphylococcus are generally found in the air.